Hello everyone, I am Dibyendu Samanta from IIT Kharagpur. So this is our third lecture on chemical and physical properties of nucleic acids. And during last lecture, we discussed about genetic material, particularly three classic experiments that proved that DNA is our genetic material, namely Griffith's experiment that proved the transforming principle or concept of transforming principle, followed by Avery, McLeod and McCarthy's experiment that clearly showed that DNA is our genetic material. Even more, Hershey and Chase experiment convincingly proved that DNA is our genetic material. Today, we will mostly discuss about DNA or nucleic acid. So here, these are the major points I would like to cover: the introduction to nucleic acids, which is our genetic material, and then building blocks of nucleic acid, followed by understanding the difference between two major nucleic acids, that is DNA and RNA, and then discovery of DNA structure and some key features, followed by some physical properties of DNA, particularly what happens to DNA if it is heated. So now, nucleic acids. So nucleic acids, like all of us, we are aware about gene. So gene is like, for example, if one kid, that kid is doing very good in academics, and there are many people in the same family, they are also very good in academic. Many times we say this is in their gene. So gene is very common to all of us, but what is the chemical properties of gene? That was not known before. So from those three classic experiments, we came to know that gene means DNA in this case. Now here particularly you can see that RNA and DNA. So RNA stands for ribonucleic acid and DNA stands for deoxyribonucleic acid. So what is nucleic acid? So as I told, that gene was known long time before, but nucleic acid also, like before establishing that DNA is our genetic material, nucleic acid was known. Almost in 1970s, 80s, during that time, scientists, they discover some acidic substances present in the nucleus. So that's why this name came, that nucleic acid, some acidic substances present in the nucleus. So then many other scientists, they actually did lot of experiments, and finally they came to know what is present inside nucleic acid. That is like all of us, we know that this is made up of nucleotides. But this way nucleic acid is first discovered. Then ribonucleic acid and deoxyribonucleic acid. Although we discuss about that DNA is our genetic material, I should tell here that RNA can be genetic material in few cases. For example, some viruses, they have RNA as their genetic material. So all of us, we know during the COVID period that whatever we are talking about, this SARS-CoV virus or coronavirus, so they have RNA as their genetic material. But this is not frequent. Only few viruses, they have RNA as their genetic material. Now we are going to discuss about little bit about the central dogma of molecular biology. This is a very important theme in molecular biology, that I also explained before, that DNA, as you can see, DNA, so this DNA goes to DNA, that means it can replicate itself, and that is called replication, followed by transcription. So DNA, whatever written inside the DNA, it can actually form another polymer of nucleotides, that is called RNA, and the process is called transcription. So here transcription is done by the major enzyme called RNA polymerase, and replication is done by DNA polymerase. Many other enzymes and factors are also involved, but those are the special enzymes that carry out this thing. And now from RNA finally we get protein. So informations are present in DNA, and proteins are actually the molecular machine which work for us. So these are known as molecular machine. They actually work for us, but the instruction present in DNA. And RNA is present in between DNA and protein. So it helps to get the protein. So this process, from where we are getting the protein from the RNA, particularly I would say in this case, we should say this is mRNA. This is messenger RNA. And it gets translated and it forms proteins. So this step is carried out by two major things. One is ribosome. So ribosome is made up of many different types of proteins as well as nucleic acid, particularly rRNA, ribosomal RNA. And also another important nucleic acids, that is tRNA or transfer RNA, that also play important role in this process. So now if we see here in ribosome, we have rRNA, ribosomal RNA. So if we see, although proteins are the molecular machine that works for us, but the thing is DNA here, RNA, which is here I am saying mRNA or messenger RNA, followed by ribosomal RNA and tRNA, all of them, they are involved to get the protein. So nucleic acids actually play important role in this, getting the protein out of the information. So it's like huge thing in our body. And we should know about nucleic acids in detail, like what are the major properties of nucleic acid. So I'm going to discuss about the very basic and fundamental thing about nucleic acid that will be applicable both for DNA as well as RNA. Wherever some differences are there, I will also explain that. Now, here I should also mention that why those terms are like replication, transcription and translation. So replication means just to make its own copy, the same thing, right? So that's why it is called replication of DNA. Now transcription means like some information present somewhere, and then just you are copying that information again. So you are writing that information in some other places, that's all. So this is transcription. So for example, we sometimes say to get the certificate or mark sheet, we say the transcript. So that is the same thing. So here DNA, this is also polymer of nucleotides. RNA, this is also polymer of nucleotides. So basically their language is same, only we are rewriting the information which is present in DNA in a different form so that it would help to make the protein, right? So after DNA to RNA, that means when the transcription is happening, three major RNA we get. One is mRNA or the messenger RNA, which is the template for protein synthesis. And then we have two different like major group of RNA also. One is ribosomal RNA and the other one is transfer RNA. So all these three RNA, they are formed from DNA. So, but all of them are polymer of nucleotides. But now from RNA we will get the protein. So protein are the polymer, so they are polymer of amino acids. So polymer of amino acids. So now if you see, the language is completely changed here. So until now DNA and RNA, they are polymer of nucleotides, but now this is polymer of amino acids. For example, if some of us, say for example, I am going to France, but I don't know French language, then what I need? I need a translator. So that person will actually, he will help me to translate the language. So similarly here, so at the end, whatever we are getting the protein, it is not made up of nucleotides. The language has been completely changed. This is the polymer of amino acids, and that's why this is called translation. So now we just discussed about the basic central dogma of the molecular biology, but there are some, updates and a little bit addition here, and I will discuss that in right place. Now, I would mostly concentrate on nucleic acids. So, those are the polymer of nucleotides. And now, if we see what is present in nucleic acids, the first thing is nucleotides. But what is what is present in nucleotides? So, in nucleotides, we have three major things. One is nitrogenous bases, and nitrogenous bases can be of different type. It can be purines, it can be pyrimidines, I will explain soon, followed by sugar. So, like pentose sugar is present in nucleic acid. So, this sugar and nitrogenous base together, so that forms nucleoside. So, sugar, that is pentose sugar. So, pentose sugar means 5-carbon sugar present in nucleic acids. So, it can be ribose or it can be deoxyribose. Ribose sugar is present in RNA or ribonucleic acid, and deoxyribose sugar is present in DNA. And nitrogenous base and sugar together, we say this is nucleoside. Now, at the beginning I told that nucleic acids, they are the polymer of nucleotides. So, now if we add phosphate group with the nucleosides, then it will be nucleotides. So, this is nucleotides. So, this is the monomer of nucleic acid. So, we will discuss more about this now. And now, RNA versus DNA, what are the major composition or differences in composition in case of DNA and RNA? So, now the major things, the major differences is the sugar group, like whatever the sugar moiety present. So, as I already mentioned that pentose sugar or the 5-carbon sugar is present in both DNA and RNA. But in case of ribonucleic acid, it is ribose. So, what is the difference between ribose and deoxyribose? So, from the name itself suggests that deoxy. So, somewhere they lack oxygen. So, if you see here, this is pentose sugar and this is the carbon number 1 prime carbon, 2 prime carbon, 3 prime, 4 prime and this 5 prime. So, this is 5-carbon sugar. And now, in the 2 prime position, you can see here OH is present. So, OH is present in case of ribose sugar, which is present in RNA. But in case of DNA, instead of OH, it should be just H. So, oxygen should not be there. So, that is why deoxyribonucleic acid. So, that is the difference. And now, this is just sugar. But now, in terms of nucleotides, if you see, we have here sugar, and then here the nitrogenous bases, it can be purine or pyrimidine, and also it has the phosphate group. So, together it will be we will say this is nucleotide. Now, I should also explain one a small thing here, but which is very important when we are discussing about DNA or RNA structure, that why I am saying that 1 prime, 2 prime, 3 prime, why not 1, 2, 3, 4, 5 something like that. The major reason is here in case of purine and pyrimidine, in that case also it has multiple carbon atom. So, just distinguish between these two, like from nitrogenous bases from purine, pyrimidine to sugar, just for the distinguished purpose, we are saying that 1 prime, 2 prime, 3 prime, so that we can discuss in a better way. So, carbon number in purine and pyrimidine should be just plain 1, 2, 3, 4, 5, something like that. Let us see in the next step what is there. So, also if you see that pyrimidine and purine, they are attached to the sugar by glycosyl bond. So, this is because the carbohydrate is attached to these nitrogenous bases. Some glycosidic group is attached to that. That is why this is called glycosyl bond. And here the phosphate is attached to the sugar by ester bond. So, ester bond. Now, about the nitrogenous bases, which are the major component apart from sugar. This is also major component present in nucleic acid. So purine, there are two, I would say there are two flavors of nucleotides present in nucleic acid. So the one flavor is purine. They are much bigger. So like adenine and guanine, they are the purine. And pyrimidine, so we have cytosine and thymine. So this adenine, guanine, cytosine and thymine, all these four nitrogenous bases present in DNA. So pyrimidines are smaller compared to the purine. But in case of RNA, we have uracil instead of thymine. So in case of DNA, we have thymine, but in case of RNA, instead of thymine, we have uracil here. But they are more or less their structure is similar, only this CH3 is missing in case of uracil, that's all. Now, if we we try to see about the nucleotide, how it looks like full chemical structure. So here you can see all these four structure, what we are showing here. This is deoxyribonucleotides. So those are the nucleotide, but these nucleotide are present in DNA. Why? Because here we don't have the OH group. See, no OH group here. So that's why this is deoxyribonucleotides, which are the major building block of DNA. Similarly, if you see here, in this case, this is also nucleotides, but this is ribonucleotides. So here you have OH group. So this is ribonucleotides, which is present in RNA. And as I already mentioned, again I am clarifying that this portion, like if I just include this portion here, so just sugar and the nitrogenous base, so that should be our nucleoside. And whenever we are including phosphate, so together I would say this is now nucleotide, building block of nucleic acid. Now, we should go into the polymeric form of nucleic acid. So we are just discussing about the monomer nucleotides. Now, if we see DNA or RNA, their basic architecture is same, that how they are getting polymerized. So if you see here in this case, so this is one phosphate group here, this is sugar, and this is the nitrogenous base. We are just writing A or adenine, just for simplicity. Otherwise, again and again we have to write very big structure. So all together, these three together, this is one nucleotide. But now if you see this is one nucleotide, I am just mentioning this is adenine, like it's attached to sugar as well as phosphate group here. Now it is next attached to another nucleotide, that is thymine. So how those two nucleotides are joined together? So that is the question now. So this is the covalent linkage between two nucleotides. Like the reason is DNA is polymer of nucleotides. So if you see in between these two nucleotides, we have one covalent linkage here, that is phosphodiester bond. So if you see, this is one ester bond and another ester bond, and it's with the phosphate group. So that's why the term is called phosphodiester, two ester bond present here, phosphodiester bond. So nucleotides are attached by phosphodiester bond to make the polymer. In addition to that, we should clarify one important thing here, that whenever we are writing any nucleic acid sequence, so for example, I am writing here A, T, G, C, A, G, C, something like that, just nucleotide sequence present in some gene or DNA. Now, this is not complete sequence. Always we refer by some direction, like I would say this is convention. On left hand side we are writing 5 prime and this is 3 prime. 5 prime to 3 prime direction. This is the sequence 5 prime A, T, G, C, A, G, C. So then what is 5 prime and 3 prime? If you see in this DNA sequence here, in this DNA sequence, at the 5 prime end, that means in the top end, the 5 prime carbon here, 5 prime carbon of this pentose sugar is attached to the phosphate group. So 5 prime carbon of this sugar is attached to the phosphate group. So DNA can extend only in this direction from 5 prime to 3 prime direction, as you can see here. So at the bottom end, if you see, the 3 prime OH is free here. So again, another nucleotide can be attached here by some reaction, which we will discuss later. So again here another phosphodiester bond can form and another nucleotide can be attached. So 5 prime end, it is capped with the phosphate group, which is attached to the 5 prime carbon of the ribose sugar, or in this case deoxyribose sugar, and the 3 prime end is free with 3 prime OH group. That's all. So this is 5 prime to 3 prime direction. Whatever I discuss about this DNA, this is true here in case of RNA also. Everything same. Only at 2 prime position, you will see OH is present here, the hydroxyl group is present here. And another difference is, like in case of RNA, we have uracil instead of thymine. That's all. Otherwise, the basic skeleton is same in case of DNA or RNA. So now, I just told the basic difference, right, in case of DNA and RNA. Although I told the basic skeleton is same, the polymerization strategy is same, but at the 2 prime position of the sugar is a major difference. The OH group present in RNA and H group is present in case of DNA, and another one is the uracil present in case of RNA and thymine is present of DNA. But this thymine uracil, that will not create lot of differences in between DNA and RNA. The major difference is the presence of the OH group at 2 prime position of the sugar. So that's why RNA is less stable than DNA. So I always told that DNA is our genetic material. So DNA is much more stable also compared to RNA. If we see RNA here, so this is like a segment of RNA we are showing here. So just we are showing 2 nucleotides here, ribonucleotides particularly. So now we have this OH group present here. And in our body, in our cellular environment, the pH is little bit basic. So pH is, I would say, around 7.2 to 7.4. So in that condition, we have excess OH minus ion in the surrounding. And from there, it will start a reaction, and that will finally hydrolyze this phosphodiester bond. So as a result of that, it is very easy that the RNA will be broken down into small segment. And that is the reason why RNA is little less stable compared to DNA. And biologically, it is very important. Now, discovering the structure of DNA, this is very important thing. Although, like at the beginning, I was, started with this, the discovery of nucleic acid. Many of us sometime, we thought that DNA was discovered by Watson and Crick, but that is not true. Like at the beginning, I told almost 70 years before the discovery of DNA structure, like Friedrich Misser, so who discovered this nucleic acid at the beginning, I told. So as a result of that, what Watson and Crick did, they discovered the 3-dimensional structure of DNA. Mostly from there we will know different physical properties of DNA. So as you can see here, in 1953, James Watson and Francis Crick, they published this paper in Nature, so describing the structure of DNA. And here you can see their model of DNA, just hand-built model, how it looks like. And because of their discovery, they got Nobel Prize in 1962, which is like, very important in terms of our biomedical science. It helps us to understand many, many aspects regarding genetics, genes and biochemistry, molecular biology, many things. So now, this is, although Watson and Crick, they got Nobel Prize because of this, and they they proposed this structure, and it was absolutely cool. But the thing is, the X-ray diffraction data, which was actually obtained by Maurice Wilkins and Rosalind Franklin, and based on that data, they understood what is the pattern in DNA. That some helical pattern is present, some basic repetitions are there. So finally, they came up with this model. And I should mention here that during the same time, there are many other scientists, they were trying to decode how DNA looks like, including Linus Pauling. So, but James Watson and Francis Crick, they came up with this model, and which proved to be true also. And now, to get more details about the discovery, this book, The Double Helix, written by James Watson. This is a fantastic book, and you can go through it. And now, I would like to focus on some key features of DNA. Like now, all of us, we know that DNA is double-stranded, DNA is double helix, all those things. But it was really difficult to discover, really difficult to discover from that X-ray diffraction data, this one, that DNA is double helix, DNA is double-stranded. So, Watson and Crick proposed that because of some pre-existing data. So, one important thing is Chargaff's rule. So, Chargaff was a big chemist. So, he contributed a lot in this, DNA, understanding on DNA structure also in that way. So, what is Chargaff's rule? So, he found that in nucleic acid, particularly in case of DNA, like I told that in case of nucleic acid, we have nitrogenous bases, purine and pyrimidine. What Chargaff found that for a particular species, for a particular cell, if you analyze DNA, what he observed that purine by pyrimidine ratio is 1. Purine to pyrimidine ratio is 1. So, this is very important thing. So, as a result of that, I mentioned that purines are those bigger structure, bigger nitrogenous bases, and pyrimidine are the smaller one. Sometimes I forget also. So, the very easy thing to remember is the short name. Purine is short name, but the structure is big. On the other hand, pyrimidine is the bigger name, but the structure is short. So, what they found that purine and pyrimidine ratio is always 1. So, that means there is some association between purine and pyrimidine. They stay together. Not only that, if we analyze even in more details, what was known that in DNA, A by T ratio equal to 1. So, A is purine here and T is pyrimidine here. Similarly, G by C equal to 1. So, G is guanosine and C is cytosine. So, as a result of that, I can say that A by T equal to 1 and G by C equal to 1 in case of DNA. So, this actually helps Watson Crick also to understand the DNA structure quickly. So, that means some specific base pairing, something is present there that is very important. Like I would say covalent structure of DNA, it was known long before. Covalent structure means that the nucleotides are attached by phosphodiester bond and the sugar is attached to nucleotide bases. So, those are the covalent structure of DNA. But this non-covalent structure, that is very important. That was not known during that time, and Watson Crick, they came up with the right model. So, in DNA, two nucleic acid strands, they are annealed, or I would say they are somehow interacting together through inter-strand hydrogen bonding. This is the non-covalent structure. And this base pairing follows the rule proposed by Watson and Crick that A base pair with T. So here A and T, they make 2 hydrogen bonds in between them, and G and C, they make 3 hydrogen bond in between them. So this is called Watson and Crick base pairing, and this is evident from even Chargaff's rule also, that purine by pyrimidine is equal to 1. And hence the 2 strands become complementary to each other. So as a result of that, what I would like to say, for example, if I say that this is the sequence of DNA A, T, C, G, C, G, T, A, then automatically I can derive the sequence of the other strand. So other strand will be T, A, G, C, G, C, A, T. Why? Because A always base pair with T, C always base pair with G, so they are complementary to each other. But only one important thing is their direction will be different. I am going to discuss soon. So this should be 3' to 5'. So DNA, those 2 strands, they are running in in opposite direction. Now this is the structure of double-stranded DNA. As you can see here, that 5' to 3' and 3' to 5'. So they are anti-parallel. And now if you see here, this is the phosphate group, this is sugar, and this is the nitrogenous bases. So that means this phosphate sugar, this is the backbone, sugar phosphate backbone, and nitrogenous bases are attached with the sugar group. And now these bases, they are forming some hydrogen bond, specific hydrogen bonding. So as I already told that adenine and thymine, they form hydrogen bonding. So they have 2 hydrogen bond here. And in case of G and C, we have 3 hydrogen bonds, as you can see in this figure. Now this is also very important. So DNA is double-stranded and helical structure, right? So as you can see, A and G, they are much bigger. So when DNA, I would like to when I will explain their structure, you will see that 2 strands are going their dimension, 2 strands are parallel. Now as a result of that, the width of the DNA should be fixed. So as a result of that, one big like adenine and one thymine, one big and one small. Similarly here, guanine is big and the cytosine is small. So as a result of that, A and G together, for example, then the width will be more, both are much bigger, and similarly T and C, both will be much smaller. So also there will be problem in hydrogen bonding. And now in terms of antiparallel, if you see in this figure, you can see this is going in this direction 5' to 3', and this is 5' to 3' in this direction. And if you see in this way, then you will be saying that this A, T, G, C, they are properly placed and they can form the hydrogen bonding. And also, during replication, I will explain what is the chemical basis of this anti-parallel nature of this DNA. And now, this is all about this, DNA double-stranded structure. And here, the helix, what was proposed by Watson and Crick, you can see here double helix. So DNA is double helix, double-stranded double helix. And what kind of helix? Right-handed helix. Right-handed helix means it's very simple. For example, if we take our right hand like this, and this thumb, this is in upward direction, and this is the imaginary axis. Now, if we take these 2 fingers, right? So these are the 2 strands of DNA. And now, if I just put these 2 fingers like this, they are rotating like this, this will be the right-handed helix, right? So DNA is right-handed helix of these 2 strands. So as you can see here, so whatever we are showing here in blue color, this is nothing but sugar phosphate backbone, and at the in between them, those nitrogenous bases, they are forming this hydrogen bonding. So this is very important. And also from their structure, they found lot of parameter. For example, in one turn, what Watson and Crick found that in one turn we have the distance is 34 angstrom, what Watson and Crick observed. But later on, little bit, defined measurement, which is more correct, that it is around 36 angstrom. And in between, in complete one turn, now we say that 10.5 residues are present. And those are, some specific details. Without knowing those, it will not be a problem. Like the major, structural property, chemical property, those are, very important thing to understand about DNA and DNA structure, how those nucleotides, they are present, and those are the instruction of life, how they are organized. And now if we see, this is just, ball and stick and space field model. So this DNA, this is the, whatever common DNA, this is the B form of DNA. This is called B DNA. Now in textbook, you might get A DNA and Z DNA. So they have some, some additional features. They are little bit different in terms of their the width of the DNA and how many nucleotide pairs present per turn. Little difference are there, but those are not important to understand the fundamentals of nucleic acids. That's all. So here, this is the last concept I'm going to discuss here: the reversible denaturation and annealing of DNA. This is very important, which is related to whatever I discuss right now. So the thing is, DNA is double helical structure, as you can see, double stranded double helix. Now, in a tube, for example, here, this is a tube and you have DNA sample here. So now, if you increase the temperature of this sample, so you are just heating this tube. So something will happen to DNA. As you can see here, the DNA double strand will be opened up. They will be coming into single stranded form. Why? Because those double stranded structure are because of the hydrogen bonding. They are non-covalent interaction. So whenever we are increasing the temperature of the solution, the hydrogen bonding will get loosened and they will be destroyed. And as a result of that, finally, you will get the single-stranded DNA, as you can see here, single-stranded DNA here. So this is called denaturation of DNA. Denaturation of DNA. So here you can see partially denatured DNA. That means the temperature is not that high yet. But if you increase the temperature, then two strands will be completely separated, and this is two strands are separated completely here. Now, again, this tube, if you again start cooling down, so again you are decreasing the temperature. So as a result of that, those two strands again will come to each other and they will form proper Watson-Crick base pairing. So that is called annealing. So again, it can go in this direction. It will form double-stranded and double helical structure. And this is very important, properties of DNA, which makes us to design many different types of experiment, which I am going to discuss sometime later. So as a result of that, this is very important thing here. And on this basis, if you see here, if I plot this, so here in x-axis, this is temperature, for example, degree centigrade. So this is, I am starting from 25 degree centigrade, and this is 35 degree centigrade, 45, all are degree centigrade, 55 degree centigrade, 65, 75, 85, 95. And this is percentage of denaturation, percentage of denaturation. Now, so I am taking this DNA sample, double-stranded DNA sample in this tube, and then if I increase the temperature, what will happen? What will be the stage of denaturation? You will see something like this curve. This is because at the beginning, at very low temperature like 25 degree, 35 degree, 45 degree, nothing will happen. So DNA will not be denatured because at that temperature, hydrogen bond will not be broken. But at higher temperature, hydrogen bond will be completely broken. So here, for example, like all hydrogen bonds are broken and now DNA, they are in their single stranded form. So that's why you can see it is not going up anymore. So this is the maximum denaturation happened. So this is called the denaturation curve. So here the maximum denaturation, and this is the 50% denaturation here. So whenever the temperature where the DNA is denatured 50%, that temperature is called Tm, or melting temperature of DNA, melting temperature of DNA. So from this melting temperature we can like comment on DNA, particularly their base composition. So this is the last slide here. So here I am showing two data set. So for example, we have two tubes and we did the same experiment whatever I explained. And from one tube, for example, from this tube, tube number A, we get this data whichever plotted in blue color, and this one, tube B, we get the data in this brown color from tube B. So what is the difference in terms of base composition, nucleotide composition in between these two DNA samples? So from this curve we can see that sample B requires more heat to get denatured. So as you can see, the Tm is higher. Tm is higher in case of sample B. Tm is higher, melting temperature is higher. This is because I would say in case of sample B, we have more GC content. G and C, like their proportion is much more compared to A. Because as I already mentioned that G and C, they have 3 hydrogen bonding. That means you need more heat, you need more energy to destabilize that hydrogen bonding. So as a result of that, in sample B, we have more GC content compared to A. So that's all. So from this experiment, we can talk about their nucleotides component. And for further reading, you can go through Leninger's Principle of Biochemistry, and this is an excellent textbook, and that's all. Thank you very much.